my project, the mitochondrial UPR regulator, ATF5, and its ability to promote intestinal barrier function through the control of the satiety response. So mitochondria are these double membrane organelles that are commonly associated with a role in energy production. But they also play a role in amino acid metabolism, innate immunity, and cellular death. And due to their essential role, mechanisms must exist to ensure mitochondrial protection. And one of these mechanisms is known as the mitochondrial unfolded protein response. This is a stress signaling pathway, and much of what's currently known has been researched in a model organism known as C. elegans. And this data suggests that the UPR is coupled with innate immunity during infection. So our question remains, is this only reserved in invertebrates, or do we have a similar association in mammals? So using an intestinal specific knockout mouse, what we do is we first looked at mitochondrial function during infection, and we see ATF5 is actually promoting mitochondrial function. We see dysfunction in oxygen consumption, lower ATP rate, increased oxidative damage, and reduced mitochondrial membrane potential. Furthermore, we next looked at histology of our intestines, both infected as well as untreated, and what we see is more damage in our knockout compared to our control. Even in the absence of infection, we see abnormalities in intestinal integrity. Furthermore, we looked at intestinal permeability, and once again, we see intestinal barrier dysfunction, as well as looking at epithelial barrier markers. And all of this is consistent with our histology. We see disruption in the barrier in our mutant compared to our wild type. Through the course of our study, we noticed something interesting. Our mutant mice were actually gaining more weight and were eating more than our control mouse. And with the association between obesity and barrier function, we next wanted to look at a transcriptomic approach to analyzing gene expression. We wanted to compare our control versus our mutant and see anything in terms of gene regulation. And in fact, we see a set of diverse genes within the intestine of our mutant compared to our knockout. Functions including things such as metabolism as well as immune signaling. So looking at this data, we see something interesting. We see a decrease in something known as CCK. And CCK is a hormone that also plays a role in satiety. So to confirm our findings, we wanted to look at CCK protein in the serum as well as transcript levels. And what we see, in fact, is lower levels in our mutant compared to our control. And through our studies, we know that obesity has an association with elevated sugar levels, also known as hyperglycemia. And hyperglycemia results in disruption of barrier function by increasing glycolysis. So if we block glycolytic flux using an inhibitor, this should hypothetically repair gut barrier function. And in fact, by giving our mutant a glycolytic inhibitor known as 2DG, we see better barrier function. So overall, we propose the following model, that ATF5 is protecting the host by maintaining the intestinal epithelium by preventing aberrant glucose metabolism through the control of the satiety response. And with that, I'd like to thank my lab as well as acknowledge our funding and Dr. Christopher Gibson that did histology as well as Dr. Netta. Thank you, and that'll be it.